morning, everyone. Welcome to the DCS webinar on biotech financing models. This morning, we had the pleasure to invite Professor Andrew Lo, Professor of MIT Sloan School of Management, and also MD, EVX, Dr. Tan Yen Chung, together with us today, sharing with you some of the insights on biotech financing models. So without further ado, let me actually invite MD of EVX, Dr. Tan Yen Chung, to give an introduction to Prof. Lo, please. Dr. Tan, please.
Thanks very much, Rachel. Good morning, everyone. Thank you for joining us today so early in the morning. I'm Yen Chung, Managing Director at EVX Ventures, the biotech VC. It is my pleasure today to host this webinar together with DCS, and also to introduce Andrew Lo. And Andrew is the Charles E. and Susan T. Harris Professor at the MIT Sloan School of Management. He is also Director of MIT's Laboratory for Financial Engineering and Principal Investigator at MIT's Computer Science and Artificial Intelligence Laboratory. Throughout his career, he has accumulated numerous accolades and awards that really showcases his contributions to his field. These awards include Battery March, Guggenheim, and Sloan Fellowships, the Paul A. Samuelson Award, the Eugene Fama Prize, the IAFE Sanghat Financial Engineer of the Year, Global Association of Risk Professionals Risk Manager of the Year, Harry M. Markowitz Award, Managed Futures Pinnacle Achievement Award, one of Time's 100 Most Influential People in the World, and last but not least, also awards for teaching excellence from both Wharton and MIT. He has also written many books, many of which are essential required readings for students and practitioners of his field, all available on Amazon if you're interested. He's also a renowned economist who has really taught and really inspired an entire generation of hedge fund quants. And the biotech financing framework that he has pioneered has inspired numerous companies like BridgeBio. And I think a lot of VC firms have also taken a lead from what Andrew has done. And in this talk, Andrew will share his insights on how financial engineering can help reduce the risk, overcome the value of death, and increase the attractiveness of biomedical innovation. So everybody, please together with me, please welcome Andrew. Andrew, the floor is all yours. Thank you.
Well, terrific. Thank you very much. I want to begin by thanking Dr. Tan, EVX Ventures, and Biotech Connection Singapore for organizing this event and inviting me to participate with all of you. And of course, I want to thank all of you for joining so early in the morning. For me, it's somewhat late in the evening, about 9 p.m. And so I want to apologize for my voice. I've had a full day of Zoom meetings, and as you can probably relate to, my voice is a little bit hoarse, but hopefully you can all hear me. So it's really a pleasure to be speaking to all of you today about a subject that I never thought I would be focusing on, which is applications of financial engineering to healthcare. By way of background, I'm a financial economist. I've had no prior training or interest in healthcare or biomedicine, and got interested in the field really because of personal reasons. A number of years ago, friends and family dealing with various kinds of cancer. And through their process and trying to be sympathetic and supportive, I realized that finance actually plays a really big role, sometimes too big a role, in biomedicine. And so what I want to talk about today is some of the work that my collaborators and I have done on thinking through financial issues, specifically as it applies to drug and device development, and maybe using some different tools of financial analysis to increase the reward, decrease the risk of these kinds of projects, and that will bring more investment capital into this important field. So let me start with my slides. I'm going to use this format, which my students seem to like because it approximates my being there with all of them. I apologize that I can't be with you in person. I've never been to Singapore, and someday I hope to visit. I think it'd be a wonderful experience based upon everything that I've heard. So I want to start with an observation that most of you know far better than I do, because a number of you in the audience are biotech experts and life scientists. Biomedicine is currently at an inflection point. And I'm going to give you 3 examples that I've come across, but I'm sure many of you have many, many other examples, and you see this all the time in your own work. The 3 examples I'm going to give you about why it is that biomedicine is at a point today like no other in history. The first example has to do, of course, with vaccine development. I think all of us are quite well aware of the fact that prior to the pandemic, the fastest time it has taken to develop a vaccine from the beginning of the identification of the pathogen to the time an FDA-approved vaccine was available, the fastest development time previously was 4 years, and that was for the mumps. In many cases, it's much longer, and in certain cases, we still today don't have a vaccine for HIV and a number of other diseases. But it's clear that this year, things have been completely changed by the mRNA technology that allowed us to develop a vaccine candidate within 42 days and within 63 days of when the SARS-CoV-2 virus was sequenced, the first patient was dosed in a clinical trial. So this is an extraordinary accomplishment that tells us the science and medicine of infectious diseases has been completely transformed. The second example has to do with these two kids, Caroline and Cole Carper. So these two kids were born with a very rare disease known as Leber's congenital amaurosis. It's a single gene mutation, a typo in their gene, that causes blindness starting from birth. It basically prevents the retina from producing an important protein that allows sight to occur. And so children that are born with Leber's congenital amaurosis begin to go blind at birth, and by the time they're 6 months old, they can't see. In 2016, a Philadelphia-based company called Spark Therapeutics developed a treatment for this particular disease. And this is what Caroline had to say. She and her brother Cole were in this clinical trial. And this is what she had to say after being dosed with this particular gene therapy. I went outside when it was snowing, and I was like, oh, I can see the snowflakes. It was really cool to actually see something that I've never seen in my life before. And what is this treatment? This treatment is a one-time injection of a virus that contains the correct version of that gene inserted into it. And the virus is injected to the back of the eye of the patient. And from a one-time injection, the corrected gene is inserted into the proper cells. And from that point forward, that particular protein is produced that eventually allows Caroline and her brother to see snowflakes and other things that I've never seen before. It's really astonishing treatment. The third example that I want to tell you about is related to a company for which I wrote a case study, a company called Agilis Biotherapeutics, based in Cambridge, just about 6 blocks away from my office at MIT. This is a company that was developing a similar gene therapy for a different disease, a disease known as L-amino acid decarboxylase deficiency. I know that's a mouthful. I had no idea what it was until I came across this company. But this is also a single gene mutation that prevents your brain from producing dopamine. Now, I suspect that most of you have heard of that neurotransmitter. It's a pretty important one. And in particular, it's important to be able to develop motor function. So infants that are born with this AADC deficiency, you don't know at first there's anything wrong. They're just lying in their crib, not doing anything. But after a few weeks, where the babies continue to lie in their cribs, not moving their arms, not moving their heads, not sitting up, not rolling over, not moving much at all, at that point, you realize something's wrong, and parents start to panic. It's a very difficult disease to diagnose, but a Taiwanese doctor by the name of Paul Hu not only understood this disease, but developed a treatment for it, very much like that blindness treatment. A virus contains the corrected version of the gene inserted into it. And in this case, the virus is injected to the brain of the infant. And I'm going to show you a video clip of patient number 4. This is a 2-year-old that was diagnosed with AADC deficiency. So what you're going to see in this video clip is the before and the after. So you'll see a baseline before the child was injected with this viral vector. And then you'll see that same child 1 year later, and then 2 years later. And remember, this is just a one-time injection, just one injection. That's it, okay? So here we go. So this is the baseline that you're going to see of the child. You see that the child can't lift his arms up, can't lift his head up by himself, has very, very little motor function. 1 year later, that same child, clearly being able to move much more than before, crawling, and he's not quite standing yet, because remember, he just learned basic motor functions starting a year ago. And so he's definitely behind, can't stand completely on his own, as you can see, he'll fall. And now 2 years later, that same child is able to stand and not quite yet ready to walk, but getting there. Now, we don't know whether he'll ever develop normally because, of course, he didn't have these important neurotransmitter when he needed it at birth. But this is just an incredible miracle. The blind shall see, the lame shall walk. You know, that's a phrase that is generally uttered in religious contexts, but it's happening today, thanks to the miracle of modern medicine. This is what I mean when I say that biomedicine is at an inflection point. Now, 3 of my colleagues, Susan Hockfield, Tyler Jacks, and Phil Sharps, published a paper, a report a few years ago titled Convergence to describe this inflection point. So in this report, they describe the convergence of the life sciences, physical sciences, and engineering. All of these are coming together in ways to develop amazing therapies for patients and their families. And the way that it is often characterized, this inflection point, is the so-called omics revolution. Genomics, the study of the sequence of our genes. Epigenomics, the study of the on-off switches that cause certain genes to be expressed and others to be suppressed. Transcriptomics, the study of how those gene sequences translate into proteins. Proteomics, the study of the 20 to 25,000 proteins that make up the human body. Metabolomics, the study of the chemical reactions in the body. And finally, most recently, microbiomics, the study of the bacterial colonies that help us with a variety of bodily functions. Every one of these omics has experienced tremendous advances over the course of the last couple of decades, with the exception of one. There is one omics that happens to be a bottleneck to all of this progress. And what is that? That's economics. And by that, I mean the fact that we have to pay for all this stuff, and the business models that we're using are not necessarily the optimal business models to be able to support biomedical innovation. In fact, it's become such a challenge that there's a name for this. It's called the Valley of Death. The idea that to get early stage breakthroughs in the science and medicine of dealing with human diseases, to get those ideas out of the laboratory and into the clinic has become harder and harder. And it's not to say that there isn't money in the field. As you know, given those of you who are in the field, there's a lot of money that's coming into biomedicine. But that money is going to later stage assets as opposed to the really early stage preclinical projects. And that's the challenge. Now, when I first heard about the Valley of Death, my question was, why? Why is there this Valley of Death? I would have normally thought that if you have a great scientific idea and you've got patients that can benefit from it, then somehow money magically appears. And it shows you how ignorant I was of the biomedical industry. It turns out that from my vantage point as an economist, the reason for this so-called valley of death is increasing risk and uncertainty. The fact that as biomedicine gets more complicated and more advanced, that actually creates greater and greater risk. And if there's one thing I can tell you about risk, it's that investors, they do not like it. So let me give you an example of that and switch to a somewhat specific example having to do with the challenge of drug development. So I suspect that all of you know that drug development is a long process. It typically begins all the way on the far left with ideas and discoveries in the laboratory. And after enough testing with Petri dishes and animal models, eventually you get to bring these ideas into the clinic with a phase 1 trial. And then if that works well, you go to a phase 2 trial, then a phase 3, and then finally you can apply for FDA approval if you make it through all of those different stages. This lengthy process actually has 3 specific characteristics that make biomedicine very difficult from the omics, the economics perspective. And the 3 features are that biomedicine is extremely costly, much more so than other fields like technology or manufacturing or retail. It's very costly to develop a proof of concept. You're talking about hundreds of millions to billions of dollars between the preclinical stage and a drug approval. Second, the probability of success is extremely low. In the area of oncology, historically, the probability of success of getting a preclinical compound into an FDA-approved drug, that process is probably historically less than 5%. And finally, the duration of that process is on the order of 5 to 15 years, depending on which area you're in and the various kinds of diseases you're trying to tackle. These 3 quantities make it very difficult for biomedical innovation to be supported by large amounts of capital, simply because investors are going to be challenged by any one of these, but for all 3 of them, it makes it very, very complicated. So let me now turn to what investors are looking for so you can get a sense of why it is that this valley of death occurs. Now, to do this, I'm going to give you a little bit of a surprise quiz, one that I've used for my first-year MBA students all the time. And I'll see how you do in this, okay? It's an investment quiz, nothing to do with healthcare. It has to do with these 4 financial assets. I'm not going to tell you what they are or over what time period they span. I'm simply going to show you what happens if you invest $1 in each of these 4 assets and you leave it there for this multi-year investment period. And I'm going to ask you to pick one of these 4 for your retirement fund or your children's college fund or for your grandparents' life savings. You can only pick one of these 4. And which one would you pick? They have very different risk-reward trade-offs, right? The green line, that turns $1 into $2. Not very rewarding, but not particularly risky. The red line, way more rewarding, $1 turns into $5, but a lot more ups and downs. The blue line, that's the most rewarding of all. It turns $1 into $8, but way, way, way more volatile, ups and downs. And the black line turns $1 into about 7, not as rewarding as the blue line, but not nearly as risky. So this is a question about risk versus reward, right? Which would you prefer? So I'll give you a few seconds to choose. Unfortunately, we're not set up to do polls. Otherwise, I would do a poll the way I do with my students, and you can see for yourself what the most popular choice is. But I know what the popular choice is. I'll tell you in a minute. So first of all, let me tell you the answer now. The time period for this exercise is from 1990 to 2008, right? That's the time period. So pretty long time period. The green line is the US Treasury bills. It's the safest asset in the world. at least for the next few weeks. We'll see what Congress decides in the United States. But assuming that we agree to pay our bills, US Treasury bills are very, very safe, virtually riskless, but they're not very rewarding. So if in 2008 you put your money in Treasury bills, well, you would have earned pretty much next to nothing over the last 12, 13 years. What about the red line? Well, the red line is the S&P 500, the US stock market. And many people feel that that is a very good investment. But when they look at this chart, for some reason, they're not excited by it. And so very few of my students will pick the red line. But if you pick the red line, congratulations, you would have done just fine over the course of the last 11 years. Okay, now what about the blue line? Well, the blue line is the single pharmaceutical company, Pfizer, one of the largest pharma companies in the world, way, way, way more risky. And if you pick that, well, once again, congratulations, you would have done even better, right? Spectacularly well. All right, what about black line? It turns out that the black line is the most popular choice among virtually every audience that I've ever applied this particular quiz to. And it turns out that the black line is the returns to the feeder fund for the Bernie Madoff Ponzi scheme, which is why I had to stop it in 2008. That's when the Ponzi scheme blew up. Now you know how the scheme got as big as it did, right? It's human nature. All of us, we are drawn to investments like this, high yielding, low volatility assets. And sometimes things that look too good to be true aren't true, like the Ponzi scheme. Now, in finance, we have a name for this type of risk-reward profile. And it's based upon the statistic known as the Sharpe ratio after the Nobel Prize-winning economist Bill Sharpe. It's basically the excess return of an investment above and beyond the risk-free rate divided by the risk as measured in this case by the standard deviation. And so using the Sharpe ratio, I can show you that Pfizer has a Sharpe ratio of 0.43. The S&P 500 is a Sharpe ratio of 0.54. And the Madoff Ponzi scheme, at least on paper, before it blew up, had a Sharpe ratio of 2.98, many times larger than the other Sharpe ratio. So that's why we are attracted to this. From a financial investments point of view, all of us would love to have high Sharpe ratio investments. And that's what we gravitate towards. Now, why am I telling you all this? It's because biomedicine, particularly the early stage investments in that valley of death, has had Sharpe ratios that have been going down over the course of the last few years. Going down because as we get smarter about treating disease, as we come up with these innovations, those innovations in turn end up making existing drugs obsolete. And that destroys value for shareholders that invested in those existing therapies. And so great advances in biomedicine, but the more you make those advances, the more complex the risk profile becomes for biomedical investment funds. And so that's what I want to talk about today. I want to talk about how to deal with these kinds of challenges. So to do that, let me give you a second investment quiz, okay? The second quiz is for you to consider the following investment opportunity. I want you to consider investing $200 million in a fund that I'm going to launch. And this fund is going to take $200 million. It's going to take me 10 years before I can return any capital to you. And the probability of my returning any capital to you at all is 5%. So 95% of the time, I'm going to return nothing, and your investment will be worthless, okay? How many of you would be willing to invest in this investment with me? Now, you know, when I ask my students this, none of them raise their hands, and I think they feel a little bad because I specifically made it personal. I said, you know, I'm going to be the one that's managing this portfolio. So how many will you be willing to invest in me? And even when the professor is asking, you know, the students say, no, we don't want this. Now, every once in a while, I'll get a brave student who will raise his or her hand and say, Professor Lowe, you didn't tell us. In the 5% cases where we are able to generate a positive payoff, what is that payoff? You see, most of the time, students don't even ask. They don't need to know, because $200 million, 10 years, 95% failure rate? No, thank you. Not interested. Well, these are the back of the envelope numbers for what it takes to develop a single anti-cancer compound. It takes about $200 million to run the clinical trials, and it takes about 10 years. And as I mentioned earlier, in the area of oncology, the historical success rate is actually a little less than 5%, a higher failure rate than what I'm assuming here. However, if in the unlikely event that you are successful, it turns out that an anti-cancer drug today will produce about $2 billion in earnings over the course of the remaining 10 years of its patent life. Why 10 years? Well, because a typical patent is for a period of 20 years. The first 10 years are taken up doing clinical trials. So you've got another 10 years left on your patent. And so you're generating revenues from years 11 through 20. That's 10 years of $2 billion a year. That sequence of cash flows is equivalent to a one-time payday of $12.3 billion in year 10 if you succeed. So let me ask you the question a different way now. Suppose I ask you to give me $200 million today, wait 10 years, and at the end of 10 years, you either get nothing with 95% probability, or you get $12.3 billion with 5% probability. Would you invest? For those of you who are quantitatively inclined, this investment is equivalent to an investment that's got a 12% annualized rate of return over the first 10-year period, but it's got a volatility of 423.5%. That's a Sharpe ratio of 0.02. And when I ask my students this, none of them want this. They're not interested. It's too risky. And that's the point. The point is that from a risk-reward trade-off, investors are not going to easily put large amounts of capital in investments like this. And this matters. It matters for patients. Patients like my mother, who was dealing with non-small cell lung cancer for a few years, and she exhausted the standard of care. So we were looking for experimental treatments for her. And I was introduced to a biotech company, a very successful one, that was developing a number of anti-cancer compounds, one of which was targeted at non-small cell lung cancer. So I met with the chief scientific officer, who brought along his chief financial officer to translate for us. And I asked him a simple question. I said, can you tell me, is your scientific agenda influenced at all by the kind of financing that you have? Because I was very interested in that scientific agenda and how they prioritize the different projects. He looked at his CFO and then turned back to me and said, influence? Our financing drives our scientific agenda. And I got to tell you, I was shocked by that response. Now, as an economist, I get it. You have to pay for stuff. But what does stock market volatility, interest rates, and Fed policy have to do with whether you should treat cancer via angiogenesis inhibitors or immunotherapy? Nothing. But in this biotech company's case, it drives their agenda. And by the way, this was a biotech company that was successful. They had a lot of money, in their company, and even in that case, their financing was driving their scientific decisions. Now, I'm no biomedical expert by any means, but isn't that backwards? Shouldn't we allow the science to be driving the financing and not the other way around. And so that's why I got involved in this field and has started to think about applying different financial technologies. And let me give you one example, specifically with regard to this pop quiz. The example has to do with what exactly is the problem here? What's the market failure? And the simple answer is that there isn't a traditional economic market failure like private goods or public goods, but that the failure has to do with the fact that the risks are too high, and we're talking about a long shot. Developing a single anti-cancer drug is a long shot, and not all investors are willing to take long shots. And so this is where we might think about using finance, because many cases, these long shots are societally important. Not only are they societally important, if you succeed, you make a lot of money. So we're not talking about charity here. We're talking about reinventing the business models by which biomedicine gets supported. And the bottom line idea is we need to figure out ways of lowering the risk or increasing the return. In other words, increasing the Sharpe ratio. That's what we need to think about doing. So I'm going to show you how to do that. Here's one way to do that I think most of you will understand just because it's really intuitive. There's nothing brilliant about this. It's basic common sense. Instead of investing in one of these anti-cancer drugs, what if we invested in 150 of them all at the same time? Now, forgive me for not addressing some of the bigger issues, like do we really have 150, and where do we get these projects? But let's assume that we have 150 of these projects. Now, granted, it's going to require that we raise $30 billion of capital. That's a lot of money. Where are you going to get $30 billion of capital, right? 150 programs, $200 million per program, that's $30 billion. Well, as an economist, I have a very simple answer. The answer is, assume we have $30 billion. Now, I know that's not a very satisfactory answer, but give me a few minutes and I'll come back to this. Suppose we had $30 billion, right? Well, then let's look at the economics. With $30 billion, the expected rate of return of 150 projects is going to be the same, 12%. I'm assuming that they're statistically independent, all right? I'll come back to that assumption later. If they are statistically independent and they have the same expected rate of return, then it turns out that when you do the math, the risk actually goes down by the square root of 150. And so now my Sharpe ratio is not 0.02, it's increased by an order of magnitude, 0.34. So if I were to ask you now, how many of you would be willing to invest in this project? Many, many more hands would go up. Why? Not charity, not because you feel sorry for me or you want to support cancer. It's because this is a better investment. It's more attractive. The expected rate of return per unit risk is higher. Now, can we really raise $30 billion? And the answer is, it depends. It depends critically on my assumptions, on the risk-reward profile of the portfolio. If my assumptions are correct, I guarantee you it is absolutely possible to raise even more than $30 billion. Why? Well, let's do a little bit more math. Suppose that I am correct, that these are statistically independent shots on goal, to use a hockey or a soccer term. If that's the case, then I argue that we can actually finance this using an instrument that really hasn't been used a lot in early stage drug discovery, and that is debt financing. We can issue cancer bonds, and that money will allow us to develop these drugs. Now, how many bonds can we issue? Let's do a little bit more math. If you have 150 projects that are independent, each with a 5% probability of success, what's the probability of getting at least 3 successes? Well, it's the simple binomial distribution. And any first-year college student taking probability and statistics, actually in Singapore, any high school student can tell you that the probability of at least 3 successes out of 150 tries, where each is a Bernoulli trial with a 5% probability, is 98.18%. You've got a 98% chance of getting at least 3 successes. Now, what are 3 successes worth? Well, we just said, a single anti-cancer drug, $12.3 billion in year 10. So 3 times 12.3 is 36.9. I can construct a portfolio for you that has a 98% chance of being worth $36.9 billion 10 years from now. So how much debt can you issue? How much money can you borrow to finance that? Well, it depends on what yields are. With a 98.18% chance of getting at least 36.9 billion, you can issue bonds up to $36.9 billion of face value, pure discount bonds. And the probability of default is 1 minus 98.18%, a little less than 2%. And at a 2% default rate, as of a couple of weeks ago, it turns out that the yield on that single A rated debt is about 1.87%. And that means that I can raise today, by issuing $36.9 billion of pure discount bonds that mature in year 10, I can raise over $30 billion today in cash from these cancer bonds. And if you allow me to use all the other tricks of the financial trade, like credit default swaps and collateralized debt obligations, I can do even better. Now, I know what you're thinking, especially the business students in the audience. You're thinking, really? You really want to do this? Because didn't those securities figure prominently in the financial crisis? And my answer is, actually, that's what gave me the idea of using these techniques. The financial crisis happened not because these techniques didn't work. It happened because these techniques, they worked way too well. These are very powerful tools for raising tremendous amounts of money over short periods of time. Now, of course, interest rates being at their near historic lows means that there's a lot of money out there available. So if we can use credit markets, if we can use fixed income securities to finance drug development, we're talking about nearly limitless resources to get over that valley of death. Now, it's true that we have to assume this IID assumption that they're not correlated. And, this is the probability curve of the likelihood of getting at least 3 successes out of 150 tries. If they're uncorrelated, then at k equal to 3, the 3 successes, you're getting 98.18%. But if they're not uncorrelated, if there's correlation among the various different successes and failures, at a 10% correlation, the probability of success of at least 3, goes down to about 89%. At a correlation of 40%, the probability of getting at least 3 successes goes down to 55. And at an 80% correlation, the probability of getting at least 3 successes goes down to about 23%. And believe me, at that level, nobody is going to want to buy your debt. It's too risky. You can't finance using fixed income securities when you got correlation. Correlation is the key. And when I told one of my pharma company friends, he said, you know what, that's what I've been telling my colleagues in the industry, but I use 3rd grade soccer as the analogy. And I told him I didn't understand because my kids, they never played soccer. And he said, well, I coach my daughter's 3rd grade soccer team. And this is what 3rd grade soccer looks like. Everybody's crowding around the ball. And you got to tell them, no, spread out. Don't go to where the ball is, go to where the ball will be. And when you compare 3rd grade soccer to World Cup soccer, you understand the difference. Now, I'm not criticizing the pharma industry. I'm not qualified to criticize them. I'm no expert in the science and medicine of what they do. But I'm telling the pharma industry from the financial perspective what they're doing, while there may be legitimate scientific reasons for crowding around an idea like immunotherapy or gene therapy, there may be financial reasons to do something else. And that's what I want to focus on, the fact that diversification can lower the cost of capital. So lots and lots of questions. I've had many of my questions come up as well that I've been trying to get answers. And the short answer to these questions is I have no idea. I'm not a biomedical expert by any means. So I've asked people in the industry whether or not these techniques could work. And they tell me that they don't know because they haven't used them. I've talked to pharmaceutical executives and they don't seem to know. And I've talked to doctors and they said, well, we understand the science and the medicine, but we're not focusing on these things. The long answer to whether or not these techniques will work is given in a series of papers that I've published over the last 8 years. I've really been thinking about these issues from the point of view of the financing, but I've been collaborating with a variety of biomedical experts. And the short answer turned into these long answers is yes, these techniques, they actually can work. For example, in terms of how much capital do we need, it depends on all these parameters. And in a recent paper that my PhD student and I published, which you can all get a copy of, we actually provide software that allows you to run Monte Carlo simulations to calculate the actual amount of money that you need in order to develop a particular set of portfolio assets. But the key to all of this is that finance and biomedical experts, they need to collaborate in order to be able to make progress. We can't do this on our own, either the scientists or the financial experts. We really need to have the science and the medicine come together, with the science driving the financing and not the financing driving the science. So in the last couple of minutes, I'll just tell you about the one final simulation, which is with orphan diseases, diseases like hemophilia, cystic fibrosis, ALS, or the diseases that I described to you at the very beginning of my talk. It turns out that the economics are actually very different. For a variety of reasons, there are many, many rare diseases out there, and then over 30 million Americans suffering from rare diseases, more than the number of people dealing with cancer, so that even though individually a rare disease may not have a lot of patients, as a group, they affect a huge number of people around the world. And for that reason, and a number of other legislative actions in the US, it turns out that you don't need $30 billion to create a really good portfolio. $300, $400, $500 million would be enough. You don't need 150 projects to get diversification. 10 or 20 are enough. And it really is because of lack of correlation. Because rare disease therapeutics in general have very little to do with each other, the success or failure of one really does seem relatively uncorrelated with the success or failure of another one. And if you do a simple calculation of how your Sharpe ratio changes when you have no correlation, it turns out that you can show that the Sharpe ratio goes up at the rate of the square root of the number of assets in your portfolio. So if you've got 25 projects in your portfolio, that means that you're going to multiply the Sharpe ratio of any individual investment by a factor of 5. And this is very attractive from the investor's point of view. So in a paper that I published in 2015 on how to construct a rare disease portfolio, it actually highlights these numbers and shows that using very conservative assumptions, you're going to generate an IRR of about 22% on a rare disease portfolio. Now, the last thing I'll mention is that that paper was read by a former student of mine who took my course many years ago. He was a chemical engineering PhD student at MIT, took my course in finance, and then left and went off and developed a very nice career in biotech venture capital. When he read that paper, he didn't realize I was working in healthcare and was kind of surprised, but he read it and called me up and said, would you be willing to redo your calculations, not with your assumptions, but with my assumptions? So I said, sure, I'd be happy to do that. And I learned a lot from talking with people in the industry. So over the course of 6 months, we ran a various number of simulations. Each one, the results kept getting better and better in terms of the investment returns, much better than 22%. And after about 6 months of these simulations, he came into my office one day and said, I just want to let you know, I quit my job yesterday. I'm going to do this. I'm going to build this company. And I have to tell you that I was scared to death because I'd never had that effect on any of my students. And so I said to him, wait a minute, are you sure you want to quit your job right now? You got a very good job. You got young kids. Maybe you want me to run a few more simulations? And then he said, no, I'm good. I'm ready to do this. And so I felt guilty enough that I invested a small amount in his company along with other friends and family. In retrospect, I wish I had put a lot more money into it because within a few months after we launched the company, other investors saw the idea behind this portfolio, and we were actually able to come out of stealth mode, raised a $40 million initial round, and then after that, another $135 million. And then shortly thereafter, a crossover round of about $300 million, and then in 2019, an IPO, and then further fundraising. And then, of course, as of a few days ago, this little tiny company that started up about 6 years ago is now worth about $9 billion. Bridge Biopharma is the company that Dr. Tan mentioned. And this is an incredible rate of return for investors. All the investors are thrilled with the outcome. But this is not what the company is proud of. What the company is proud of is that in a little less than 6 years, the company has developed a pipeline of 21 assets, of which 4 of them are in phase 3 clinical trials. And actually, of those 4, over the course of the last few weeks, 2 of them have received FDA approval. And so now this little tiny company has 2 approved drugs. And patients that would have died if it weren't for these therapeutics are now going to live. That's the amazing thing. So this brings me to my conclusion, which is that all of this I mentioned is really personal for me. And it became even more personal when I came across the story of my colleague, and many of you know him, Dr. Harvey Lodish, who is a cell biologist at the MIT Whitehead Institute. When I came across Harvey's story, I decided right then and there, I want to be Harvey Lodish. I want to be him. And let me tell you why. In 1983, Harvey was approached by a venture capitalist asking if he would be willing to collaborate with him to create a drug for patients that have a rare disease called Gaucher's syndrome. So Gaucher is a single gene mutation that prevents your body from producing an important housekeeping enzyme. And without that enzyme, fatty acids build up in your spleen, your bone marrow. And by the time you reach your teenage years, you're in a lot of pain, in a wheelchair, and most of them die, at that age. So this is a really serious disease. And from 1983 to 1991, Harvey and his colleagues, they actually ended up developing a therapy that was approved called Cerezyme, the very first enzyme replacement therapy. And since that time, that therapy and many others like it have saved literally hundreds of thousands of lives. And in 2011, the company was acquired by Sanofi for about $20 billion. You might have heard of Harvey's little company. It's called Genzyme. And $20 billion is not bad for a biologist. But that's not why I want to be Harvey Lodish, although that's not a bad reason. I want to be Harvey because of what happened in 2002. You see, in that year, Harvey's daughter became pregnant with her second child, Harvey and his wife's second grandchild, a boy named Andrew. Great name, by the way. Andrew was diagnosed in utero with Gaucher's syndrome. What are the chances of that happening? So, I asked Harvey this. It was a very emotional conversation. I asked Harvey, in 2002, you found out, but did you know back in 1983 that you had this mutation? And Harvey said, I had no idea. I was, interested in the science, and I thought if I could do some cool science and help patients, that would be great. And he reminded me, 1983 was 20 years before the human genome was sequenced. So nobody had any idea whether or not they were a carrier unless they had it in the family and that they would know. And, he said that I never in a million years would have expected that that I would be one day saving the life of my as yet unborn grandchild. So when Andrew turned 10 years old, he developed the full-blown symptoms of Gaucher's, but he's doing just fine thanks to the drug that Grandpa developed. This is why I want to be Harvey Lodish. I don't have an MD or a PhD in molecular biology. I will never be able to develop a drug that will save my as yet unborn grandchildren. But then I realized something. I realized that, finance doesn't have to be a zero-sum game. We can actually all be Harvey Lodish if we invest in the drugs that will save our as yet unborn grandchildren in the future. With the right kind of financing, at the right scale, it is actually possible to do well by doing good. That's the wonderful thing about biotech. Sorry, I teach MBA students, so I have to have some sound effects. So that's one of the reasons why I'm so excited to be with all of you today. Many of you here in the audience, you are the future of the biotech industry. And I'm hoping that all of you will join me and other people in the financial industry so that we can all become Harvey Lodishes of the future. Thanks, and happy to take questions and comments.
Thank you, Prof. Lo. That was a very interesting and also inspiring story shared. So we have gathered a few questions from the audience, and I'd also like to thank your insights through the talk. The first question is that, you mentioned about correlations among rare diseases and how that's impact the Sharpe ratios. But what happens if there are any other factors external to this, just correlations such as regional differences of the ecosystem, market access and all that? How would that affect your investment decisions?
Yeah, that's a really important set of questions. And this is exactly why you need the modeling software as well as collaborations between finance and biomedicine. Every one of those considerations affects the outcomes. So you have to incorporate all of these differences, all of these different features in your simulation to come up with as realistic a business model as you can. I mean, that's what Neil and I were. Neil Kumar was the founder of BridgeBio and currently the CEO. That's what we were doing for those 5 or 6 months. We were changing the assumptions about what it costs to develop a drug, changing the probability of success estimates, changing the number of clinical trials that were going to be financed over a period of time. All of those are things that need to be incorporated into the analysis. But they can be done. It's doable. And so using the tools of modern financial engineering, we can actually develop insights into those kinds of risks and rewards. And once you do, and you present it to investors, you will then be able to get the kind of funding that you need in order to be able to develop your therapeutics.
Thank you, Prof. Lo. And there's a second question saying, like, one of the example has given earlier that, if you're needing a 30 billion to start a fund, why not focus on companies that are already past phase 2, since the probability of success for those startup companies in phase 3 is actually larger than 50% actually.
That's right. Yeah. So in fact, that's exactly what big pharmaceutical companies do. So big pharma companies generally focus on not doing a lot of internal R&D. Of course, they do some. But over the course of the last couple of decades, big pharma has been doing less and less R&D and more M&A. So what big pharma companies will do is to look for assets that are in phase 2 trials. And after they complete phase 2, then they'll go in and pay a big amount of money to acquire those and in-license those assets and then develop and market them. That's the problem with the valley of death. You see, when you do that, that's great if you're in phase 2 or phase 3. But what do you do about preclinical assets? That, in my view, is where the benefit of financial engineering can be the greatest. So while I'm glad that there's money in the later stages of drug development, and thanks to big pharma, they're very actively pursuing these phase 2 assets, I want to be able to figure out how to raise money for preclinical, for the startup companies, the seed companies that are ultimately going to turn into those big successes 5 or 10 years from now.
Thank you, Prof. Lo. The follow-up question. So are you aware that any other VC firms, either in the US or other regions, is actually employing your models, to choose their portfolios, and how does the models actually affect their decisions?
Well, so, there are actually a number of VCs that have been employing this model. I wouldn't want to call it my model because, portfolio theory is certainly not unique to me. I may be, the one who's applied it most directly, to healthcare. But venture capitalists have been doing this since the very beginning, right? VCs, they pick multiple companies. They don't invest in just one. So VCs already understand this. What I guess I'm adding to this particular discussion is that the typical VC fund is often too small a scale. And VCs, I think, need to be bigger. They need to have more assets and they need to have more companies, mainly because biotech is a more risky and larger financing needs business. When you think about getting diversification in the tech world, a tech company can be launched for a few $100,000. And with a few $1 million, you can get proof of concept for an app for social media or for other kinds of online retail type of platforms. With biotech, a few $1 million is maybe what you need in the seed round, and you don't get proof of concept until you're spending hundreds of millions of dollars. So I would say that VCs already know this idea, but a number of them recently have started raising larger and larger amounts of funding. One good example is Flagship, the VC that created Moderna. Their most recent round for their latest fund is over $3 billion. So that's an example, and there are many other companies that are taking to heart this notion that you need scale in order to reduce risk, increase the likelihood of reward, and get investors excited in your portfolio.
Thank you, Prof. Lo. I think we'll entertain one last question. This is actually also from the audience. You mentioned that you have made a lot of assumptions in your model, and you keep changing those assumptions to adjust to different ecosystems and markets. So what are the key difference for Asian Pacific markets that you think you should be adjusting for those assumptions? And just to highlight to the audience a few key indicators, please.
Yeah, so that's a really great question, particularly for this audience. I would say that there are maybe 2 or 3 really important differences between the US markets and Asian markets where you would want to adjust your simulation parameters. One is that most Asian countries have single payer healthcare systems. And what that means is that drug prices are much more tightly controlled by governments than in the US. And what that means is that the revenue assumptions are going to be lower in Asian countries in general. On the other hand, you could also keep in mind that in countries like China, where the population is just really large, or India, the patient population being so large means that you're actually going to be able to conduct clinical trials in some cases more quickly. For example, in rare disease therapeutics, diseases that are rare or ultra rare in the US may not be nearly as rare in Asia, because simply the size of the population will allow you to identify more candidates. So I would say that the population size and the cost of, or the revenue assumptions of these therapies need to be adjusted to deal with those differences. But otherwise, I would say that they're very, very comparable. The talent pool in the US is significant, but the talent pool in Asia is equally significant, in my opinion. Things that are happening now are happening, in biotech, are happening around the world. As I showed you in the case of AADC deficiency, the pioneering work was done not in the US, but it was done in Taiwan by Dr. Hu, who is at National Taiwan University. So there's talent in biotech, all over the world.
Thank you, Prof. Lo, very much for your insights and your time sharing with our audience and your interesting and inspiring speech. And thank you, EVX, for organizing this webinar with us. And thank you, everybody, for the audience and your time this early morning on Wednesday, and also Prof. Lo at evening times. So have a great day. Does Yuan Chong would like to say a final word?
No, I just want to thank everybody. Thank Andrew for being here, and also thank all our participants for once again waking up early in the morning, and for Andrew for being here until 10 p.m. at night. So I really appreciate it. It's been very illuminating, and on behalf of everybody here in Singapore, I'd like to thank you for a great talk.
Yes, Prof. Lo. Look forward to future talks from you too. All right. Have a nice day, everyone. Bye.
Bye-bye.
Thank you.
Thank you.
Bye.
Bye.